Okay, today I will discuss uh, another fossil pteridophyte plant of your concerned syllabus, that is Calamites. As you know very well, that the Calamites is a very known fossil plants which occur in during the Carboniferous period and Triassic period of Paleozoic era. As you know very well, in last lecture, I have taught you your very interesting one fossil fern, which is belongs to the same division, Stenophyta, that was Stenophyllum. Today, I want to discuss your next fossil plant of your syllabus, that is the Calamites. First of all, I discuss with you the classification, the classification, the classification of your fossil fern, Calamites. If you are talking about the classification of this plant, it belongs to group Pteridophyta because these are the characters of the plants belongs to the Pteridophyta group. So it belongs to the group of Pteridophyta. And as you know very well, that the division of this fossil fern is Stenophyta. As you know very well, there is some specific diagnostic features uh, in division Stenophyta are occur in Stenophyllum as well as Calamites. The presence of groove and ridge in the stem due to composition of silica, due to decomposition of silica towards the uh, upper layer of the epidermis, the stem become the differentiated into ridge and groove. They have very specific uh, extraordinary vascular system in stem with the carinal canal and vallecular canal in stem, and the sperm is bifringillate. The sperm is bifringillated, and they are so good. They are so good indicator for ecological decay. They are indicator for presence of silica and gold in the soil, and they are also indicating the member of the division Spenophyta also indicated that they grow where there silica and gold are abundant in the soil, and they also indicated the fertility of the soil. So it is a excellent bioindicator group of the plants which belongs to division Spenophyta. And the commonly name of the member of this division are horsetail plant because of the aerial shoot of these plants is very similar to the horsetail. So they are known as horsetail plant. They are belongs to subdivision Spenopsida, and they also belongs to order Calamites. It's come under the family Calamitaceae, and genus is Calamites. As you know very well, that the the plant the plant which occur in a fossil form are not completely found. Many paleobotanists work on fossil plant, and they find out that the plant which occur in a fossil form are not completely found. They occur in fragments. There are a lot of research work going on continuously at NBRI, Lucknow, National Botanical Research Institute, Lucknow. It is a very famous center in India, uh, in which a lot of the paleobotanists work on continuously on a fossil plant. Like that, the Calamites are also found in fragments. Some paleobotanists work on the rhizome of the Calamites, some work on the leaves of the Calamites, and some work on the branches pattern of the Calamites. Today, we discuss the reconstructed plants. It means the plant which not completely found in the fossil form. The Calamites, the Calamites are found in fragment in the fragment of rhizome, in the fragment of aerial shoot, the ring of branches. The genus Calamites uh, appeared in Upper Devonian, were most abundant during the Carboniferous and become extinct early in the Triassic period. It the Calamites plant dominant during the Upper Devonian period, mostly the Triassic and Carboniferous period of the Paleozoic era. According to some paleobotanists who work morphology on the morphology of Calamites, the plants was a tall tree and it is about 20 to 30 meters tall. The tree uh, the was present with well-developed rhizome, aerial shoot, and a ring of leaves. If you see the picture, first of all, you have to see the picture of this fossil specimen. This is the specimens of the Calamites. According to paleobotanists, the Calamites, the plant of Calamites was tall, which having a well-developed underground portion of the stem in the form of rhizome. If you see the rhizome clearly, you can see that the rhizome is divided into node and internode, and from the node, according to paleobotanists, from the node towards lower side, towards the soil surface, a bunch of adventitious root are developed which function as absorption of water and minerals. And from nodes of the rhizome towards upper side, there is a develop also of roots, a group of roots. And from the horizontal rhizome towards upper side, there is a develop aerial shoot. Aerial shoot. The aerial shoot, if you see the picture of aerial shoot of the Calamites, it is also divided into node and internode, and from the node of the aerial shoot on a both side, both lateral side, there is a developed lateral branches. There is a developed very frequent lateral branches, and the lateral branches of the Calamites, according to paleobotanists, it is also divided into node and internode. It is also divided as node and internode. At the node of lateral branches, there is a develop a ring of leaves, a ring of leaves. The leaves was lanceolatus to speculate in shape and size. This is the external morphology, yeah, reconstructed plants of the Calamites, which worked by the paleobotanist. This plant were dominant during the Triassic and Carboniferous period of Paleozoic era. If you are talking about the external morphology of the plant, the rhizome were differentiated into node and internode. I have already discussed with you when you see the diagram. From node develop many adventitious root towards the lower side, and the main function of adventitious root is absorption of water and minerals for proper growth and development of the plant. Aerial branches were or was, you can see there is a was and were are used for fossil plant. Aerial branches were born on the upper side of the rhizome. These were generally constructed at the place where these joined the rhizome. The lowermost node of the innate branches produce a ring of adventitious root. Because the root of the fern is not well developed, they are so primary developed root, adventitious root. It is very similar to monocot plants of angiosperm. The aerial branches were highly articulated, as you see, as you seen in picture. If you are talking about the leaves were developed in whorls, rings at the upper node. These also had branches borne in a pair. Each node had a single whorls of leaves. The leaves were smaller and perhaps were photosynthetic organ. It, it has been uh, observed by some paleobotanists after seeing the specimen of Calamites that there is no large leaves were developed in on the branches of the Calamites. It is considered by paleobotanists that the leaves were small and they are so main photosynthetic they organ of the plants. The leaves were lanceolate, linear, and spatulate, spoon-like, with a single vein. There is no lateral vein present in the Calamites. According to paleobotanists, that the leaves were simple, lanceolate, linear, and spoon-like. Genus Calamites is divided into subgenera on the basis of mode of branching and general growth pattern. So, according to some paleobotanists, it has been discovered that the genus, on the basis of the mode of branches, how the branches develop on the node, and what is the growth pattern of the node of the lateral branching on the node. It has been discovered that the genus Calamites divided into subgenera. First subgenera is Euclamites. According to this subgenera, it has only one to many branches. It is a diagnostic features of this subgenera of the Calamites, one to many branches at the node. The subgenera which has one to many branches at the node come under this subgenera of the genus Calamites. Second is Calamitina. Branches in this subgenera, branches were in rings which were separated by a series of branchless nodes. This is the diagnostic features of the Calamitina. And third one is diploclamites, in which in this subgenera two to three branches, produced with branches alternating from node to node. This is the diagnostic feature. This is the diagnostic features of this subgenera of genus Calamites. And fourth one is Cruci Cruciclamites. In this subgenera, there were large number of branches produced at each node. I hope this specimen of Calamites come under Cruciclamites. If you see the picture, you can see that on each node of the Calamites there is a developed large number of lateral branches. Large number of lateral branches. And last one is styloclamites. In this subgenera, there were few irregularly scattered lateral branches are developed, and in some in some species of Calamites, it has been observed that in this category of subgenera, sometimes the plants were without branches. The plants were without branches. If branches are developed, they are developed irregularly, scattered. They are not forming a regular pattern. And if you are talking about the internal structure of Calamites, according to paleobotanists, there is no complete information available about the internal structure. But according to some paleobotanists, the little bit information we have Calamites about the stem. About the stem. In TS of stem, it has been observed that the outermost layer of stem is epidermis. It is a single layer and protective layer of the stem, and just below the epidermis there is a large large region are present in the form of cortex. The cells of the cortex were thin-walled and it was parenchymatous in nature, and it is considered by paleobotanists that these parenchymatous cells of the cortex act as storage of excess food material itself. Below the cortex, in the center, in the TS of the stem, in the center, there were pith were present, which surrounded by a ring of primary vascular bundle. It has been observed by some paleobotanists that a carinal canal, it is a diagnostic features of the member of which belongs to Sphenophyta, division Sphenophyta. It is a presence in the region of the vascular system. The presence of carinal canal in a vascular region and vallecular canal in the cortex region is the diagnostic features of the member which belongs to division Sphenophyta. So this this character represent a very important symbol of Calamites, which is very similar to the other member of the division, Sphenophyta. A carinal canal, which representing the protoplasm, were present in each vascular bundle. The carinal canal represent act as protoxylem. It is a primary xylem. The metaxylem is centrifugal towards the center and was made of pitted tracheids. So the conduction of water and minerals were take place by these pitted tracheids. In group of the pteridophytes, the xylem is made of only one element, that is tracheids, which act as conduction of water and mineral properly. This is the information about the Calamites which we have in fossil plant. In fossil plant, we haven't a lot of information about the plant. So this is the finishing of your very important genus of your syllabus, which belongs to division Sphenophyta. Thank you very much. If you have any question, you may ask me, please, without any hesitation. As you know very well about fossil plant, we have a lot of information about the development of the archegonium, development of the anthridium, and about the fertilization, about the embryogenesis. We have knowledge about the external morphology. भाई complete हो गया ना? हाँ जी.